Hi, I'm Jennifer Gerton from the Stowers Institute in Kansas City. I'd like to thank the organizers for this opportunity to present our research, and also to just say this has been a very stimulating meeting. I don't get to think about genomic medicine very often, and it's really been great to listen to everyone here. So I'm a chromosome biologist. And what I want to understand is how the genetic material gets passed from cell to cell, from parent to child, and then over the course of evolutionary time as well. What I'm going to tell you about today is a story of how a particular chromosome fusion forms in the human genome, what it tells us about the human karyotype, and what it tells us about chromosome evolution, and a little bit about what makes our karyotype uniquely human. So I've really had the pleasure to work with a dream team of collaborators on this project. My lab over here on the left, Tamara and Leo are really experts in chromosome biology, analysis of repetitive DNAs and their evolution. We've had the fortune to work with Adam Philippi and his group, which they are fantastic at genome assembly. And then also Eric Garrison and his group, who are experts in developing innovative methods to analyze multiple genomes at once, called pangenomics. As many of you have already noted, we are really in this very new and exciting time for human genomics. We know the first genome assembled back in 2002, as was so beautifully presented by Eric Green. It was a milestone achievement. It's been incredibly important for our understanding of human biology, but there were parts of it that were missing, about 8%. And now, thanks to long-read sequencing and the work of these large consortiums, we really have a complete human genome with chromosomes assembled from telomere to telomere. I've had the fortune to work with these consortiums on several projects, and I just want to set the stage a little bit for what I'm going to tell you, because there were things we had to learn before we could get to where we are now. Back in 2020, Glennis and Karen Miga each led efforts in the first assembly of a human centromere, these repetitive sequences. Followed by a couple years later by the assembly of complete human genome, followed by now multiple human genomes, and even more recently, we've got the assembly of primate genomes. And those really allow us to do some evolutionary comparisons that are very enlightening about the human genome. But there are parts of the genome that we historically have not had access to. They've not been in any of the reference genomes, and sometimes we refer to this as the dark matter. And those regions are highlighted in the red and the green probes on this human karyotype. So this 8% of the genome is still a big challenge. Actually, it's still quite challenging to do these assemblies. You saw the red regions, the centromere assemblies that Glennis has been able to achieve. This is really at the very cutting edge of what's possible. But they're still quite challenging. And my lab has been building on these new sequence assemblies in order to understand the structure and the function of these regions of the genome. And we think that these regions of the genome are quite interesting and represent a new frontier, because repetitive DNAs are often unstable. They often drive recombination events. And so I think there are lots of things left to discover about the human genome once we start really investigating these newly accessible parts. And so the story I'm going to tell you about today is about a structurally variant chromosome in the human genome called a Robertsonian chromosome. This is a type of chromosome that was actually first noted by William Robertson over 100 years ago, and he noted these types of chromosomes in grasshoppers. So these types of chromosomes are actually very widely observed across plants and animals and insects, so really across much of the tree of life. What is a Robertsonian chromosome? It's a chromosome that is the fusion of two chromosomes. So it's when the centromeres are near the ends of the chromosome, And these are often described as either acrocentric or they can be telocentric chromosomes. And the chromosome literally fuses together in some way that we don't understand to make a single metacentric chromosome. This is a major mechanism of karyotype evolution, and it's a major mechanism of speciation. So the mouse, it turns out, is a rock star when it comes to these types of chromosome evolution events, Robertsonian translocations. So actually over 100 different karyomorphs have been documented in the house mouse. And these can create a reproductive barrier. So I'm just going to show you one or two examples of these 100 distinct karyomorphs that have been reported. So one population of house mice might have 20 pairs of telocentric chromosomes, and another population of house mice might have 11 pairs of chromosomes. And this is the same species, but this creates a reproductive barrier. We really wanted to understand why these chromosomes form so frequently, broadly in nature. And we decided to take advantage of all these new resources in the human genome to ask this question for humans. Why do these translocations form so frequently? So what's frequent in the human genome? They exist in about 1 in 800 individuals. And again, we decided to partner with the Philippi and the Garrison groups to ask this question. And we reasoned that if we could understand this in why and how these happen in the human genome, we might be able to understand why they happen more broadly in nature, and it might tell us something about chromosome evolution more generally. So we want to answer this question today in humans. And now I want to tell you about the specific chromosomes in the human genome that are acrocentrics. The people who are cytogeneticists in the room already know this, but there are 5 chromosomes in the human genome that are acrocentric chromosomes. They're the ones shown in these green boxes. These are the ones I've really come to know and love: 13, 14, 15, 21, and 22. It turns out that most Robertsonian chromosomes, not all, but most in the human genome, involve chromosome 14. And in this particular karyotype, what I'm showing you is an example of where one copy of chromosome 21 is fused to chromosome 14. The other really common fusion is the 13-14 fusion. But 85% involve chromosome 14. Most Robertsonians form in female meiosis, and again, they're relatively common. About 1 in 800 individuals has one of these translocations. So how do you find out that you have one of these translocations? A lot of times people find out because they're having trouble getting pregnant, and they go get their karyotype done, and then they find out that they have one of these fusion chromosomes. So there can be fertility and subfertility associated with these types of fusion events. The 14-21 fusion event actually can sometimes underlie cases of Down syndrome, and in particular, it's the most common cause of age-independent Down syndrome. And I've got an example karyotype of that here, where there's 2 normal copies of chromosome 21, but also a copy of chromosome 21 that's fused to 14. And so this karyotype came from an individual who has Down syndrome. We also know that these can underlie a different trisomy, that trisomy 13. They're often associated, the 13-14 fusions. And then there was this paper that found this association between Robertsonian chromosomes and cancer incidence, but the mechanism for this is just completely mysterious and unknown. So, going back to this image of chromosomes, I think I want to highlight that the P arms of the acrocentric chromosomes, essentially where those fusions are happening, were not sequences that were present in any of our reference genomes before 2022. We were missing the sequences on those 5 chromosome arms. And I can tell you what's there is just a lot of repetitive DNA, tons of different satellite DNAs, undiscovered satellite DNAs, as well as the ribosomal DNA repeats. And so we know this now thanks in large part to the work from Adam Philippi's group, who painstakingly assembled these regions of the chromosomes. And then Adam Garrison's group came in and started looking at these regions of the genome across the human population. Now, we don't have a lot of these assemblies, but he wanted to understand sort of what was in these parts of the genome. And he and his postdoc, Andrea, shared their findings with me, and it's something that I find completely astonishing. Okay, so this was their finding. The short arms of these 5 chromosomes contain very large, that is megabase size, blocks of identity. So what I'm depicting with this cartoon with these bands of the darker color are these regions of near identity that exist between these 5 chromosomes. Before this finding, the only part of the genome known to have these large blocks of identity were the PAR regions, so the pseudo-autosomal regions. Those are the regions on the X and the Y chromosome that cross over in meiosis. But now we have these 5 heterologous chromosomes that have these regions of near identity. And I got really excited because I knew, based on the cytogenetic literature, that somewhere between the centromeres in yellow and the rDNA regions in gray was where these translocations were thought to happen. So we were very excited. We were exchanging a lot of ideas, and eventually what we came up with was a model for these pseudo-homolog regions and how they might lead to Robertsonian translocation. So this was the term they gave these regions of homology, pseudo-homolog, because it's like they're homologs, but they're heterologs. So the model is that these regions on 2 different chromosomes can line up with each other in meiosis, undergo a crossover recombination. That would fuse the long arms of these chromosomes, leaving these little bits of chromosomes that would be lost, and that would be our Robertsonian event. I told you in the human genome there are 3 chromosomes in particular that tend to form the Robertsonians, 13, 14, and 21. So what's special about these 3 chromosomes? Well, it turns out that they have a subregion of shared homology that is quite special. It has 2 special properties. The first is that it is inverted on chromosome 14. And the second is that this subregion has a repetitive DNA that I believe is creating a recombination hotspot. So I'm going to tell you a little bit more about this potential recombination hotspot. This subregion of homology contains this repetitive DNA called SST1. This is a repeat, again, that we discovered in the 2022 genome. Actually, it was known before this in gibbons, but had not been studied much in the human genome. And what Leo, postdoc in the lab, did was he extracted all the monomers of this repeat, and he made this phylogenetic tree so we could look at the relationship between the different monomers. And what he realized was that there was a special version of this repeat that was shared specifically between these 3 chromosomes that form the Robertsonians. And we named this subfamily 1. And it's recognizable because it has this large internal deletion of about a kilobase. And it also has this very special binding site. It has a predicted binding site for PRDM9. PRDM9 is a meiotic gene, and the protein can create a local recombination hotspot based on previously published work. So putting this all together, we came up with this model that these regions of homology are maintained through ongoing recombination that can occur in meiosis. The reason these chromosomes can get together in meiosis and exchange information is that they share these ribosomal DNA repeats. This is going to put them together in 3D space because they can co-localize within the same nucleolus. It's going to bring these adjacent sequences near each other. they can undergo meiotic recombination. Crossover recombination is a bit more rare than other types of recombination in meiosis, but in the case where chromosome 14 lines up with chromosome 13 or 21 using its inverted homology, and a crossover event happens, that would then fuse these two chromosomes together. So that was the working model, that was the hypothesis. How are we going to test it? We decided to test it by choosing 3 cell lines that had Robertsonian chromosomes and fully assembling those chromosomes. So here we used cytogenetics and paints to validate these 3 different Robertsonian chromosomes, 2 13-14 fusions and a 14-21 fusion. We were able to, and the model sort of has 3 predictions. We're going to have this repetitive DNA at the breakpoint. All the rDNA from these fused chromosomes is going to be lost because it ends up on these little bits. And there's going to be 2 centromeres on these Robertsonian chromosomes. So we collected a whole bunch of different data. I'm not going to tell you about all the different experiments we did, but the long-read data helped Adam's lab assemble these Robertsonian chromosomes from telomere to telomere. And what I can tell you is that these 3 predictions were true in all 3 cases. So it seems like, at least for now, this model holds. This was very exciting because it was the first time that Robertsonian chromosomes had been assembled in any genome, and certainly in the human genome. And this work was published last month and got picked up by the Washington Post, so that was a nice bonus for us. So we also think that this general kind of some of the themes of how these chromosomes fuse together can also tell us about how they form more generally out in nature. And so I just want to point out what we think are some of the more generalizable features of this model. This idea that chromosomes can have shared and inverted homology, that the chromosomes have to get together in 3D space. This could be through any kind of clustering, telomere clustering, centromere clustering, and then of course you're going to require some crossover recombination. So I want to just finish by kind of again zooming out and thinking about what this tells us more generally about chromosome evolution, and I want to use the primates, the new primate genomes, to illustrate this point. So from the Robertsonian events, we thought that it's actually really important to understand which chromosomes can find each other in 3D space. So knowing which chromosomes have these ribosomal DNA repeats may actually be quite important for understanding chromosome evolution and chromosome interactions. So here I'm showing you the same 5 chromosomes in the human genome that have the rDNA, which is highlighted with the green fish probe. We did the same thing for these primate genomes that were recently published. And again, these are still gaps in all of these genomes. We can't assemble across these regions. But I think something, when you put them all together this way, something that becomes immediately obvious is there are, in the orangutan, there's 10 chromosomes that can have rDNA, and in the gorilla, there's only 2 chromosomes that have rDNA, and the chimps and the bonobos have 5 chromosomes with rDNA, but it's not the same 5 as the human. So this is a really structurally, rapidly evolving part of chromosomes. So that was pretty interesting to us. And through some work with Adam that I don't have time to go into, we further realized that this orangutan, the 10 chromosomes with rDNA, that actually represents the ancestral state. And so sort of all these other configurations that I'm showing you here have derived from this state based on a series of structural rearrangements that gave rise to this. So to just say that in another way, these 10 chromosomes have rDNA in the orangutan species, but then there are a number of chromosomes, the ones in light blue, that have lost their rDNA repeats. However, when you lose your rDNA repeats, it turns out you don't lose all that other repetitive DNA, you don't lose all that heterochromatin. So these light blue chromosomes have all of these sort of vestiges of once having had rDNA. And it turns out that that can still impact how the chromosome works and how it behaves. And we're working on that for a lot of these light blue chromosomes to kind of understand what those sequence relics mean. But just to give you a very quick example, I'm going to tell you about human chromosome 9, and then I'm going to sum things up here. So human chromosome 9, it has been known, again, in the cytogenetic literature for a long time that there is a high number of structural variations. So 8% of the population, you can have an addition of a heterochromatin band, you can have the loss of a heterochromatin band, and then there's this inversion that can happen in about 1.5% of the human population, and it happens at a similar level as these Robertsonians. And here's actually an image of a chromosome that has one of these inversions. So you can see this very large block of heterochromatin that switches its position relative to the centromere marker. So this is one of these inversion, chromosome 9 inversion events. And so we were doing some sequence analysis of chromosome 9, and now we know that chromosome 9 used to have rDNA. Okay, so now we are kind of looking at these events through a little bit of a different lens and through this lens of how the Robertsonians occur. And it turns out that chromosome 9, like the acrocentric chromosomes, has these PHR regions. It has pseudo-homolog regions, and it has 3 of them, and they contain this highly recombinogenic version of SST1. And so our hypothesis, our working model, that this is an ongoing project in the lab, is that we think that some of these structural events that have been observed are probably being mediated by these sequences that are sequence relics from this chromosome having once been an rDNA chromosome. So kind of knowing where your chromosomes came from might be able to tell you something about sort of their evolution and where they're headed. Okay, so we think that uncovering the molecular mechanism of how these form kind of solves this centuries-old observation, and it's an example of what we can learn with more complete genomes. But we also think that having access to these repetitive regions of the genomes is going to reveal mechanisms of genome stability, both in health and disease, and we're really starting to appreciate the significance of these repetitive DNAs as a driver of karyotype evolution and potentially speciation. So I will thank my wonderful collaborators, Adam Philippi, Eric Garrison, and members of their lab. I thank my lab, thank the technology centers at the Stowers Institute and our funding sources, and I'll thank you for your attention, and if there's any clarifying questions, I'll take those.